Welcome to my channel. Here I talk about my science career, job hunting experiences and insights and how they could help you. I deliver presentations related to pharma, documentation, quality and microbiology. And I also discuss how what I know can help you either as an employer, a job seeker, a peer or a potential collaborator. Through this channel I hope to show others I know my stuff and also improve on my communication. Perhaps this will inspire you to do something similar or help you in your career. Hi there, I'm Paul Yeatman, a Bachelor of Science graduate and an experienced microbiologist of over 15 years seeking to continue my career after a redundancy, be that in the lab, in production, in quality or other. As well as here, I can be found on LinkedIn, Twitter and my microbiology blog, so be sure to check those sites out for more information on what I can offer you. Today I will discuss Welcome to what makes an objectionable organism. I first developed this presentation in 2006 as part of a series of 12 training packages to be delivered to my team each month. Recently, I updated the training to include current references and to keep it relevant. I hope you find this beneficial. Who am I and what is my industry experience? I have 15 years of microbiology and pharma experience, comprising 9 years in sterile injectable manufacturing, environmental monitoring, with about 5 of those on the bench, 2 in QA and the rest leading teams. 6 years working in non-sterile manufacturer as a team leader. I controlled the aseptic media fill program for 2 years when I worked at the sterile injectable plant. I performed autoclave loading pattern validations, incubator validations and qualifications, computer validations, as well as validated many test methods and conducted many total microbial count and total yeast and mold counts. What is in this training for you? If you work as a microbiologist where you test total microbial aerobic counts or TMACs and total yeast and mold counts or TYMCs, and for specific microbes, and in roles where you investigate the nature of microbes and what impact they may have on the product and the end user, this presentation will benefit you. It is important to make the distinction between specified microbes and objectionable microbes, as they are not the same thing. This presentation will provide you with the following knowledge. What makes a specified organism? What makes an objectionable organism? How to determine if a microbe is objectionable. Things to consider when you conduct a risk assessment on this. And benefits of knowing the differences between specified and objectionable organisms. Presentation aims and outcomes. When testing non-sterile products, as well as allowed numbers of microbes, there are certain types of microbes that are specified in the regulations as not allowed, and still others that can be determined to be objectionable. And this presentation will deliver all this knowledge to you in the following ways. You will find out what makes a specified organism, what makes an objectionable organism, how to determine if a microbe is objectionable, some risk assessment considerations, and finally the benefits of knowing the differences between specified and objectionable organisms. So pretty much how that helps you, your company and your investigator process out. What is a specified organism? This is an organism that, according to the pharmacopeia, cannot be present in a sample, which the usual amount we test is 10 grams. These microbes are specified either as they may be an indicator organism or they may actually pose a product safety concern. The USP 1111 microbiological examination of non-sterile products lists several organisms which, depending on the route of administration, can be objectionable in a drug. These are Staphylococcus aureus, Escherichia coli, Pseudomonas aeruginosa, Candida albicans, and bile-tolerant gram-negative bacteria. And because these are actually stated in the relevant monographs, they are known as specified microbes, so they're not allowed in the product. For example, Candida may be specified for a vaginal application route. Escherichia coli and Salmonella could be specified when the product is ingested because there's a capability to cause disease here. Pseudomonads and Staphylococcus aureus might be specifically stated as not allowed for topically applied products because rubbing such microbes on your skin may cause disease. And E. coli, though capable of causing disease on its own, is typically used as an indicator organism. So the presence of this microbe could mean the product or sample has been contaminated with fecal matter, or it might just mean that E. coli is there. And as well as these stated microbes, any other microbe recovered from your testing might be objectionable. So that's why that's important to make the distinction between specified microbe and objectionable microbe. What is an objectionable organism as opposed to a specified organism? An objectionable organism is one that we do not want to see in our product based on what the microbe is, so its genus and species. How many microbes were recovered? Is it above the limit? Is it a certain amount that is known to cause disease or be capable of causing disease? How the product is used, so is it topical, is it breathed in, is it ingested, who the end user is, so is that typically a healthy person, a sick person, an immunocompromised person, things like this. And I would argue that a specified microbe is not necessarily objectionable. It's specifically stated as not being allowed. Any microbe recovered from any total microbial count test should be identified and examined to determine if it is indeed objectionable. And the PDA defines objectionable organism as microorganisms that due to their numbers and pathogenicity can cause infection, allergic response or toxemia in patients receiving the product. The PDA also notes that where the product appearance, stability or efficacy can be affected by the microbe, then it's objectionable. So basically you don't want your microbe to either damage the patient or to actually damage the product. How do we determine if a recovered microbe is objectionable? A non-sterile manufacturer should be concerned if microbes that are not specified are recovered during the specified test, and they should also determine if microbes recovered by your total aerobic microbial count and total yeast and mod count tests are potentially objectionable. And you can evaluate this by reviewing such things as the microbe pathogenicity, so what diseases is the microbe capable of causing, and might this microbe in the product cause disease when applied. The microbe source, where is the microbe found in nature and in what numbers, so versus how many are going to be in the actual product. What is the likelihood of this microbe in a product causing disease in a patient who may be immunocompromised? So that's another consideration. If the numbers of the microbes are lower than what you might find in nature, would they normally get into a patient, and if so, would they cause a problem if that patient has a problem? Potential product spoilage. Can the microbe by its very nature spoil the product or inactivate it? Might the microbe use the product or its components as food? Might the microbe produce substances that are toxic or deactivate the active ingredients in a product? So what I mean here is you've got a microbe, the microbe utilizes the product, it produces byproducts. Some byproducts can be toxic, so that can cause a problem with the patient, and some byproducts can actually deactivate the active ingredient of the product, which renders the product ineffective. Results of your count testing. Was the specified microbe recovered during the TAMC or TYMC test? The specific test is meant to revive microbes that might not be able to be recovered via the general count test. If you recover Staph aureus in the TAMC test and you find none of it in your specific test for that microbe, you'd probably want to investigate why that is the case. Was the total aerobic microbial count test contaminated, for example, or was the specified test conducted correctly? And finally, the end user and the mode of application. Is the end user sick or healthy? Was a potentially pathogenic skin microbe found in a topical cream? For, say, Ventolin, was Burkholderia capacia or a pseudomonad found in an inhaled product? Another example would be for products given to cystic fibrosis patients. You don't want to let them inhale a pseudomonad or a Burkholderia because that's probably going to cause problems in their lungs. And if this is the case, there's a potential to infect your end users, so you need to investigate it. Risk assessment considerations. Most of this is mentioned on the previous slide, or at least a very similar variant of it. A lot of what we do is based on risk, how comfortable we are with risk, the risk to the company, the risk to the product, and the risk to your customers. And when you recover a microbe, your training and experience will give you a good idea if the microbe might cause issues. Here, as part of your risk assessment and investigation, you need to consider such things as recovered microbe count. Is the count above your alert or action level? Are the number of microbes likely to cause product spoilage? Preservatives and antimicrobials in a product. You would consider the preservatives and antimicrobials in the product with an expansion on the last slide, and in combination with the numbers and types of microbes recovered, they could proliferate in the product, causing spoilage by using up or deactivating the preservatives. They could also just give the product a colour tinge, which would then make them objectionable based on what the TGA advises. It is also possible that a high number of microbes could result in reduced potency of an antimicrobial product. So again, the microbe is objectionable because it's altered your product. You would want to look at the validation data. Were the microbes recoverable during the product's validation studies? If they weren't and you found them during testing, that could cause you concern because you'd have to wonder how the microbes got there. It's unlikely to be in the actual product if the product is shown to be toxic to those microbes. So you'd want to be looking at a breakdown in process, and you want to find out where the contamination came from. You would look at the intended use of the product and where the recovered microbe typically causes disease, as I discussed on the previous slide. And also, I would advise you look at the historical data of the product. Here, an examination of historical data can show what is typically recovered and incapable of being recovered in a product. So that would get back to the validation data that I mentioned. If you suddenly start recovering a genus or species that has never been recovered in that test ever, it may represent a loss of control in your process, something amiss with the product or starting materials, or even something more interesting. A brand new microbe in your product is certainly notable, and it might be objectionable. An examination of historical data is also important from a risk perspective, as it will give you an indication of the likelihood of undetected contamination in past batches, and also the likelihood or potential for contamination in currently on hold batches or batches that are scheduled for production. What are the benefits of knowing the differences between specified and objectionable organisms? Well, basically, if you recover a microbe that is not a specified microbe, be it from your specified microbe testing, your total aerobic microbial count, or total yeast and mold count testing, based on the microbe, how many there are, your product, and the intended user, the microbe might be objectionable. And as the slide says, being aware that any microbe recovered during TMAC or TYMC testing is potentially objectionable. It is important, as people tend to assume that specified equals objectionable, and that's not the case. Potentially any microbe in your sample is objectionable. By examining microbes recovered during your aerobic count testing, one may determine if they are detrimental to the product or patient. This ensures that when we release product, they are quality products, and they do not have unexpected, in brackets, negative impacts to us or the customer, be that dead customer or failed business. In conclusion, you will now know the following: what a specified organism is, what an objectionable organism is, how to determine if a microbe is in fact objectionable, some things to consider as part of your risk assessment, and how all of the above can benefit you. So basically, a specified microbe is one where a monograph specifically requires testing for it, and objectionable is a microbe based on what it is, the product and the end user, that can potentially cause harm to either the product or the end user. Here I refer you back to slides 6 and 7 for the full details of how to determine how objectionable you find a microbe and the things you should consider as part of your risk assessment. Knowing the difference between specified and objectionable microbes and how to determine what is objectionable can help you develop processes to document how you determine such things, how to best conduct your investigation, and also improve the speed and efficiency of such investigations. Such knowledge will also help you make the correct decisions or justified decisions, so when you are audited you have acceptable evidence and answers that are not cited for lack of documentation, process, or failure to adequately investigate an issue, or for producing adulterated product. I will provide a PDF link to this presentation in the video notes, and you can always rewind to something you would like to hear again. I should note that me presenting on this topic does not mean you can take this and train your staff based off it. If you would like to do that, I strongly encourage you to employ me in such a role, or better still, give me a job in your lab, production or QA department so I can use my skills and knowledge to help you and your staff on a long-term permanent basis. I hope you have found this presentation informative, and I would like to thank you for listening. The last slide, which I'll leave on screen for about 15 seconds, provides references and additional reading. If you have found this video interesting or informative, please subscribe, like, and if you are really keen, share it with others. If you have something to say about this video, or perhaps a suggestion for a future topic, please leave a comment. Until next time, this has been Paul Yeatman, a scientist seeking career satisfaction. Kapla!